tutorial on an introduction to the microscope. You watch this on your own and you come into class prepared and ready to show me what you know. We'll talk about the care of a microscope, what parts there are, naming so that when I instruct you to do something with it you know what I'm talking about, and then also how to focus. That's the most important piece I think for our classes, is how to focus the microscope. Let's begin by looking at just speaking a little bit about the care of a microscope. Always carry with two hands. That's really critical. You don't want to drop one of these. They're rather expensive pieces of equipment. When you're cleaning the lenses, please only use lens paper. Don't use scratchy paper towels or anything else. It's really important not to scratch the lenses. Really critical is to not force the knobs. If the knobs feel like they need to be forced, if the focusing knobs are stuck, that usually means something might break if you force them past that point, so don't do that. Always store them covered to keep dust out. I do have these very fancy grocery bags to cover the microscopes with. That helps keep the parts clean and requires less maintenance in the future. When you plug in your microscope, make sure you keep objects clear of the desk and the cords. So sometimes people even plug in on the opposite side of a lab bench so that the cord isn't hanging down. It's really easy to get caught in a cord and potentially pull a microscope off of the lab bench. That's not a safe thing either. Next, let's talk about some of the parts of a microscope. We'll start from the top. You've got an eyepiece. That's where your eye goes. That's the part you look into. Next we have the body tube. That's the piece that connects the eyepiece with the rest of the objectives. The revolving nose piece holds multiple lenses. Ours will generally have 3. Our microscopes are parfocal in this class as well, so that means if you switch from low to higher power or high to lower power, it should stay reasonably focused. You might need to do a little fine-tuning, but parfocal means that you can switch between the lenses and they stay focused, so that's very helpful. An objective lens, or just a lens, or just an objective, it's called all of those, are those magnifiers attached to the revolving nose piece, and those are what allow you to look at the specimen in higher magnification. The arm is a carrying piece, also holds the objectives up over the stage. The stage is the next piece. That is the piece of the microscope on which the specimen rests, and then you can view it through the objective. The stage clips are the parts that hold a slide or a specimen onto the stage for viewing purposes. The coarse focus knob is the bigger knob on the side of the microscope. This is what moves the stage up and down relatively fast and allows you to focus. It changes the focal length between the lens and the specimen, allowing you to get a clear picture. That's really important not to use when you're on the highest power objective, but you use it for a lot of other things. Additionally, we have a fine focus knob. Sometimes they're within one another. You'll have the coarse focus on the outside and the fine focus one on the middle, but in this particular case they're separate. So the fine focus knob is one that moves the stage much more slowly and allows you to really get more detail on those higher magnifications. The base of the microscope is what holds everything up. Also usually includes a toggle switch for on and off. The diaphragm is an adjustable kind of circular piece that you can turn around and switch between allowing more or less light through the hole in the stage and then hence through the lens and up through the eyepiece. So your obviously beams of light are what you're looking at. And the light source is down here. I'll get to that in a second. But the diaphragm is what limits or extends how much light you're allowing through. And then in our case, we have electrical microscopes. We use regular electric light for this. When I was little, I had a little mirror down there on my microscope, my personal microscope, and you could reflect light up through here, which, it was a similar concept, but not quite the same. We have more reliable light source this way. Next, we'll look at the different objectives in a little bit of detail, or the lenses. The smallest one on your revolving nose piece is the scanning lens, or low power. It's usually about 4 times magnification. The medium-sized one is a low power lens, about 10 times magnification. The longest objective is a high power lens, and it's usually about 40 times magnification. And sometimes there will be an oil immersion lens, and that's at 100 times magnification. Something to keep in mind is that the eyepiece itself has a lens in it, and that already has a magnification of 10 times. So you need to multiply whichever the case may be. So for example, if you are on the scanning lens, it's got a 4 times power magnification, and then the eyepiece in addition has 10 times. That means it's 40 times magnification between these two, and then 400 between these two, and then 1000 between these two. How do you focus? I've got some skills that I learned over many years of microscopy. First, obviously, you need to put your specimen on the slide on the microscope. Use the stage clips. And then you actually should make sure that the nose piece is on the lowest power before you even put the slide on, but I have this as tip 3, that's okay. So use the shortest nose piece, shortest objective on the nose piece, and what you'll do is you'll use the coarse focus then once you've got your specimen on the stage to bring the stage up as far as it possibly can go until it stops, and don't force it any more than that, it'll stop naturally. From that point you'll look into the eyepiece and you'll use the coarse focus to move the stage slowly downward to find your object. This is the best way to find what's on your slide, because if you have to move it up or down, you have so many more options for trying to find it. If you have to just only move it down, you will bump into what you're trying to see at some point. Oftentimes the slides you'll be using are stained, so you'll see them pink or kind of a turquoise green color. And then once you've found what you think you're going to be looking at, you need to use your fine focus to get more detail out of it. And then again, our microscopes are parfocal, so if you need to switch to a higher power objective, which is usually the case, once you have it focused well on the scanning lens, you can switch it over to a higher power lens really easily, and it should stay reasonably focused. So again, kind of repeating, follow the steps to focus using low power. Make sure you always focus on low before high. It makes things a lot easier. Then you click the nose piece to the longest objective. Again, do not use the coarse focus knob on high power, only on low power. That's really important. The fine focus is what you use when you're on high power. And use the fine focus knob anytime you want to look at something. Here we've got some lovely plankton. Just a picture. Just wanted to sneak something in on you guys to make sure you watched. I would like you to be able to tell me who this is and which one of these guys you might use a microscope to see. I'd like to speak about a couple different types of microscopy that are available. We have, in addition to the light microscope that I showed you, which we'll be using in class, there exists things like a scanning electron microscope that makes scanning electron micrographs. This is a picture of one. It feeds a beam of electrons into this chamber and bounces them off of the specimen, and it comes back and it's detected by a computer, and the computer then forms an image from that. The image might look something like this. This is a tardigrade, also called a water bear, little tiny bug, little tiny arthropod, and you can see a lot of detail of the surface of this creature. So it can give you fine detail of the surface of an object. Scanning electron micrographs can. On the other hand, we have something called a transmission electron microscope. So if you want to look through something and see what's inside, you would need to use this. And this is what one looks like. In addition, similar to the scanning electron microscope, the transmission electron microscope also uses beams of electrons, but instead of bouncing them off the surface, it allows them to transmit through. And from that you might get something like this. This is Chlamydomonas, single-celled organism, and this micrograph shows quite a bit of internal structure of this single cell. You can see a lot of the details. Think about a disadvantage you might have to using a transmission electron microscope as opposed to a scanning electron, and vice versa. So with scanning electron you can look at the whole organism and even a live organism and bounce it off the surface, whereas in a transmission electron micrograph you need to take a fine slice of that organism or whatever you're trying to look at and transmit through it, so you by definition can't look at anything that's alive. We'll be using regular light microscopes, and here's, for example, a beautiful micrograph of just a picture, a microscopic picture of a pine needle. So you can see a lot of details in there, and that's the sort of microscopy we'll be doing in this class. So these are your questions. This is basically your assignment. There are only 3 questions. So from slide 7, go back there and check it out. Who is this and which one of these might you need a microscope to see? Hopefully you remember that. Question 2, compare the scanning electron micrograph image with the transmission electron micrograph image, and then what can you see and what can you not see in each of these, and then how does the light micrograph image compare to the scanning electron micrograph and TEM images. So answer those 3 questions, bring them to class in 2 days, and I hope you enjoyed this tutorial. Have a great day.